Okay, so day two. I shouldn't say day two, but I was supposed to do this yesterday, but turns out that wasn't going to happen. But what I want to do right now is go over this paper called Diagnosis of Alzheimer's Disease based on disease-specific autoantibody profiles in human sera. And I want to go over this one as well called Autoantibodies Reacting with Vasopressin and Oxytocin in relation to cortisol secretion in mild and moderate depression. So I'll start with this one first, and I'm not actually going to go through the entire papers. I'm just going to go through, basically the abstracts, and then I'm also going to, for this one, I'm just going to go over the abstract, the introduction, and then I'll probably just go to the discussion. So regardless, autoantibodies reacting with vasopressin and oxytocin in relation to cortisol secretion in mild and moderate depression. So what is vasopressin and oxytocin? So let's do a quick Google search on those. And so vasopressin is a peptide hormone, and it's also called antidiuretic hormone, so ADH. So as I said before, we like having multiple names for the same freaking thing in biology, and that sucks. So it makes it very difficult to keep track with all the chemicals, especially when you have 2 to 5 names for the same chemical. Anyways, it's also, as we said, it's also called ADH, and it's also called AVP, so, or argapressin. So there's 4 names already for this one chemical. So it's a hormone, and it's synthesized as a peptide prohormone. So a lot of hormones in your body, especially the peptide hormones, aren't made in their final form. They're usually synthesized as a full protein, and then those proteins get proteolytic cleaved into different hormones. And then those hormones are secreted, captured, and how do I phrase that? They're regulated so that they are released when they need to be released, but they aren't created in their final active forms. Anyways, vasopressin is a hormone synthesized in neurons in the hypothalamus and is converted to the AVP, which is vasopressin. So it's originally created as the prohormone and in the neurons, and then it is converted into the final AVP form. And then it travels down the axon, which is the long part of the nerve cell. It travels down the axon of that cell, which terminates in the posterior pituitary and is released. Let me just make sure. Yeah, so the posterior pituitary, and is released from vesicles into circulation in response to extracellular fluid. So these chemicals are made in the hypothalamus, and then they are transported along their axons into the pituitary gland, where they are stored in vesicles or little bubbles within the cells of the pituitary, and then given particular signaling, it will then be released, and it'll be released in order to help. Basically, if your blood is too concentrated in salts, or if it has too much osmolality to it, then or hyperosmolality is another term for that. Because of that, it'll convince the body to hold on to more water to make sure that you're not too salty. So that is what vasopressin does. And I'm trying to think if there's another. We should go through all the roles. So second, AVP, I should go through the 3 roles of it. So first, it increases the amount of solute-free water, so water that doesn't have any salts or sugars dissolved into it, and increases the amount that is absorbed back into the circulatory system from the filtrate in the kidney tubules of the nephrons. So the nephrons, or the kidneys, have liquid that it has filtered out of the blood. Now, what you would want to do, especially if you're, say, getting dehydrated, is you'd want to absorb some of that water back out without the waste products or without the salt. So this is one of the hormones that can initiate that process. Anyways, the second one is the constriction of the arterioles, which increases the peripheral vascular resistance and raises arterial blood pressure. So an arteriole is a small diameter blood vessel in the microcirculation that extends and branches out of an artery and leads to capillaries. So it restricts those, which is increasing blood pressure to a degree. And a third, and I think a possible, I wouldn't be able to tell you exactly the medical implications of that. My presumption would be that if you are getting dehydrated, that means your blood is getting thicker, and so you might want to increase blood pressure to help the blood keep on flowing, even though it's a little bit more viscous. That's my presumption. If you actually know, leave it down in the comments. And third function is possible. So some of this vasopressin may be released directly into the brain from the hypothalamus and may play an important role in social behavior, sexual motivation, and pair bonding. So this might be a social regulator or a chemical that might be released so that you form strong bonds with people or creatures, whatever. So pair bonding. So it might play an important role in feeling good to help bond with something. So that's vasopressin. And oxytocin will have a very similar effect as well. So oxytocin is another peptide hormone, and it's a neuropeptide, which would be a peptide that is used by neurons to communicate with each other, which vasopressin might also be a neuropeptide as well. And it is normally produced in the hypothalamus and released by the posterior pituitary. And it plays a role in social bonding, sexual reproduction, and childbirth, and the period after childbirth. So I think that's all that we really need to know about oxytocin. Just so that you are aware, vasopressin and oxytocin, these are two important hormones that are produced in the hypothalamic tissue and then gets put into the pituitary tissue. And so they both have similar modes of being produced and similar modes of being secreted. And also, what else should I say? They also play possibly an important role in social interactions. So if you could imagine, your psychology is very often dictated by cellular signaling and chemicals interacting, or I should say signaling to other cells what the heck is going on. And so one of the important aspects of that is the brain training you to do things that are going to be beneficial for you. That would be something that would be evolutionarily advantageous. So in regards to forming very good social structures and good communities, these hormones would play an important role in positively reinforcing you to do things that will help strengthen those social connections, if that makes sense. So if you could imagine, I should also mention that the method by which these two chemicals, these two peptides might cause that is through cortisol secretion. So cortisol is ironically a stress hormone. It is a hormone that is released after you undergo some stress. So let me look this up just so that you are all aware of what's going on. So cortisol, and I should type in Wikipedia. It's also called hydrocortisone. So sometimes if you have, I should say, autoimmune conditions or inflammation on the skin, you can also be prescribed, or you can get hydrocortisone cream, which would help modulate the inflammation process. But it is a steroid hormone, and so if you can look at this photo right here, or this chemical structure right here, this is in the general structure of steroids, which are derived usually from cholesterol or a cholesterol-like molecule. So, and you can see here they have a lot of carbons and hydrogens with very few polar groups on them. And so this is a very fatty and also a very stiff molecule, like most other steroid and steroid hormones. So it's in the class of glucocorticoids, and when it's used as a medication, it's also known as hydrocortisone. And let's see if there's anything else that I think the most important thing is knowing that it plays many different functions in the body, and frequently it is released following stress, so such as working out, or I would presume after studying or working very hard, you might get a release of cortisol after it, and that would be one of the feel-good hormones, because the release of cortisol actually does feel good. So if you could imagine, if this process gets dysregulated, either with the vasopressin and oxytocin level, or maybe if it gets dysregulated on the cortisol level, then if someone does something that would normally bring them joy, but they don't end up getting joy from the thing that is supposed to cause joy, if you could imagine, that would end up resulting in depression or the lack of motivation, because your body frequently will release feel-good hormones so that you positively reinforce good behaviors, or behaviors that end up making you more likely to thrive in society. So if you think about this, if any of those processes, if any of those positive reinforcement mechanisms get dysregulated, then you end up having basically a lack of motivation. And if you could think about something, if someone is depressed and you can't understand why they're not feeling better, and you tell them just, it's all in your head, or just think happier thoughts, well, for someone whose biochemistry is working properly, that is the solution. The solution is to think happier thoughts and to do things that make you feel better. But if you are depressed and if you have your chemicals are out of whack, then they do something that is supposed to make them feel better, and then they don't get the joy from that thing. And if you don't get the joy from doing something, it becomes more and more difficult to make a habit out of joy-causing actions. So there would be, it's not to say that it is completely useless to do those joy creation, how do I phrase that? It's not completely useless to try to do those things over and over again to try to help with your joy, but just know that the amount of chemical release that they are getting out of those behaviors are going to be stunted in comparison to a healthy-minded person, unfortunately. So this is where some people would argue for the use of chemical modulators or psychiatry in order to help get over those humps. But yeah, depression is quite a, it can really screw up with people's ability to be happy in things that are supposed to make them happy. So I've been rambling. Let's go to the background then. So abnormal vasopressin and oxytocin signaling may contribute to the altered activity of the hypothalamo-pituitary-adrenal axis, or the HPA. So remember, this HPA, your hypothalamus, your pituitary gland, and your adrenal glands, these play a huge role in your hormone regulation. So remember, for both vasopressin and oxytocin, the hypothalamus is what produced the proteins, and that gets turned into the final peptide hormones, and those get transported to the pituitary where they are stored, and then they are released. And often those released hormones are going to act upon the adrenal glands, for instance with cortisol release. If I remember correctly, I should look this up just to make sure I'm not pulling your legs, but no, it's not hypothalamus, it's the adrenal glands. I believe cortisol is production. I'll just look it up on Wikipedia. I should have looked this up before. It's produced mainly in animals, mainly by the zona fasciculata of the fasciculata. I don't know how to pronounce it, but of the adrenal cortex. So yeah, part of the, hence the name Kourt. So Kourt, it means that it's coming from the cortex. So, or at least is involved in the cortex in some way. So signaling may contribute to the altered activity of the hypothalamo-pituitary-adrenal axis, or the HPA axis, in major depression. However, the underlying mechanisms remain unclear. This study characterized plasma levels and affinities of oxytocin and vasopressin reactive autoantibodies, so auto-ABSs, in relation to disease severity, and plasma cortisol response to physical exercise in patients with mild and moderate depression and healthy controls. So, methods. Physical exercise was used to elicit plasma cortisol response. So remember what I said. Cortisol is a stress hormone, and running and exercise is a form of stress that you can put your body on, but you feel good after it, and that is due to a combination of different chemicals, but cortisol is one of them. And this is done in 23 male patients, so unfortunately not done in women. It'd be nice to have a woman cohort as well, but you know, there's so many things that you can do. Continuing with depression and 20 healthy controls, and plasma samples were obtained before and after exercise. So just before the exercise, patients and controls were evaluated by the Montgomery and Osberg Depression Rating Scale, or MATTERS, and you don't need to care too much about this, but this is just a way to quantify with different qualities that people have. It's a way to quantify the severity of their depression. That way you can use that as a scale or use it as a variable in your tests. Anyways, they did this and divided them according to depression severity. So there were 14 mild cases and 19 moderate cases. So these are the 23 male patients. Plasma levels of total and free vasopressin and oxytocin reactive IgG, IgA, and IgM autoantibodies. And remember, IgG antibodies are the long-term antibodies that are usually produced later following an infection. IgMs are the ones that are usually produced right away, and IgAs are often antibodies that are produced as a result of intestinal, and I haven't read this, I haven't verified this, but I think also possibly in the respiratory tract as well. But continuing, just so you know that they did this examination on multiple types of antibodies. And they were measured by ELISA, which is just a, how do I phrase that? It's a method of quantifying whether or not you have antibodies in a sample. And if you have antibodies that are reactive to a particular protein in your sample. Anyways, they were measured by ELISA, and affinity and IgG and IgM autoantibodies were measured by plasmon resonance techniques at baseline before the exercise and analyzed with relation to MATTERS and cortisol response. So this is just a plasmon resonance techniques. I'm not very familiar with it. Just know that it is a way to quantify how strongly these antibodies are reacting with the protein of choice. Because one might make the argument or make the hypothesis that perhaps the stronger the antibodies are interacting with the protein, perhaps the severity of the disease might increase. Anyways, immunohistochemistry was used to evaluate autoantibodies binding to the rat hypothalamus. So immunohistochemistry would be using antibodies to help, in this case, to help visualize the localization of antibodies in a tissue sample for microscopy. Anyways, let's continue. Results: the plasma levels of oxytocin and vasopressin reactive total IgG autoantibodies, were lower in patients with moderate depression versus controls. So the levels of the autoantibodies were actually lower in the depressed patients. So what could cause that? And patients with mild depression. So as people actually had less total IgG autoantibodies against oxytocin and vasopressin if they had severe depression. So that would normally go against the what we would initially believe, right? But let's continue. Plasma levels of both oxytocin and vasopressin-free IgG autoantibodies were negatively controlled with MATTER scores. So And remember, these are plasma levels of both the oxytocin and vasopressin free, so ones without binding to oxytocin and vasopressin. Continuing, affinity values for IgG and IgM autoantibodies for both oxytocin and vasopressin displayed 100-fold variability among patients or controls, but no significant group differences were found. So the affinity, or the strength at which the antibodies are binding to oxytocin and vasopressin, that doesn't seem to be having any effect as well. Patients with moderate depression displayed blunted response to cortisol secretion to physical exercise. So this is interesting. So the patients with moderate depression, and keep in mind the moderate depression is the worst case. So moderate, there would be a more severe case as well, but moderate and mild depression, moderate is the higher version. So moderate depression, patients with moderate depression displayed blunted responses of cortisol secretion to physical exercise. So basically, what is happening is the moderately depressed people are exercising and they are not releasing cortisol, which they should. They should be releasing cortisol because the release of cortisol is going to make them feel better after it. And baseline levels of vasopressin, total IgG, and IgM autoantibodies correlated negatively, and the vasopressin-free IgG autoantibodies correlated positively with plasma cortisol after physical exercises. So baseline levels of vasopressin, total IgG, and IgM autoantibodies correlated negatively. So these correlated negatively with plasma cortisol. Oh, I should see. So the baseline levels of IgG and IgM autoantibodies correlated negatively with plasma cortisol after physical exercise. So the higher the baseline levels of these total antibodies were correlated negatively with it. So if they had more of it, then they had less of a release of cortisol. And vasopressin-free IgG autoantibodies correlated positively with the plasma cortisol levels after physical exercise. So let me look at this real quick just to make sure I'm fully understanding it. I'm not having a good time understanding what exactly the vasopressin, like the free versions of it are. So I'm assuming the antibodies that are not bound to vasopressin, I don't know if they're supposed to and just aren't binding to them, or if that's just any other antibody in solution. I'm guessing it is the vasopressin, the ones that are supposed to bind to vasopressin but aren't for some reason. Those are negatively, I should say, they're positively associated with plasma cortisol after physical exercise. So if they're not binding to the vasopressin, then apparently they are releasing more cortisol, which is closer to what a normal person experiences. So yeah, that does make sense. So immunostaining of magnocellular hypothalamic neurons of the supraoptic and paraventricular nuclei by plasma IgGs was present in 30% of depression and 14% of the control groups, but the staining was not abolished by plasma preabsorption with oxytocin or vasopressin peptides. So let me look at this. Immunostaining of I apologize, I won't be able to parse this for y'all, but if you can understand that the first read through, go right ahead. I'm trying to get through these quickly, so I won't have time to, really look into all the techniques. Anyways, these data show the changes of levels but not affinity of oxytocin and vasopressin reactive autoantibodies can be associated with the altered mood in subjects with moderate depression, and that levels of the vasopressin reactive autoantibodies are associated with cortisol secretion. So they didn't go as far to say that it is, they're saying the levels of the oxytocin and vasopressin reactive autoantibodies, those levels are associated with depression, but the absolute strength of them is not. Now, what are some of my what would the word be? Some of my critiques of this is, are they just dealing with the autoantibodies, or are they also dealing with the anti-idiotypic autoantibodies as well? Because with the previous paper that I went over in my last video, in regards to narcolepsy and cataplexy, it wasn't just the autoantibodies they had to worry about, but it was also the antibodies that are against those autoantibodies that might also be playing a role in the progression of disease. And I also don't know if whatever technique they used to test for these autoantibodies, I don't know if they were able to remove the complexes, because there are immune complexes of the autoantibodies and the anti-autoantibodies that end up sequestering each other, and then they sometimes do not show up in typical immunity, or I should say in typical ELISA, or typical techniques to find these antibodies. So that would be a very interesting thing to look at as well, and I wish they did. Because if you could imagine, if there was an anti-antibody, then that anti-antibody might look very similar to vasopressin and or oxytocin. And if that is the case, those anti-antibodies might be inhibiting the effects of vasopressin and oxytocin as well. We just don't know. So that is good information to have. So with that, I'm going to move on to the next one. So the diagnosis of Alzheimer's disease based on disease-specific autoantibody profiles in human sera. So I'll just go over the abstract, but this is important to talk about because we can actually use the concentration of these autoantibodies as a biomarker for different diseases. And in this particular paper, they used the presence of or I shouldn't say they used the presence of autoantibodies, they actually screened for autoantibodies for a laundry list of different proteins in the body. And they came up with a set of them that are very closely associated with Alzheimer's. So this would be similar to how we train, say, an artificial intelligence to look at biomarkers and then give an output, which is a diagnosis of what disease that person has. So we do the same thing in science as well, where we will screen for different biomarkers, or we will screen for different chemicals in the body, and then if there is an abnormality or a change from baseline, an alteration from what the normal human or the normal healthy human has, then we can say, okay, is this positively correlated with a certain disease or some diseases, or is it negatively correlated with certain diseases? And then if we get a large enough sample of, or a large enough distribution of different biomarkers, we can then use those biomarkers as a way to diagnose diseases and diagnose pathologies. So in this particular paper, they used those biomarkers to actually make a patent, which was very effective in diagnosing Alzheimer's. And what's great about that is then you have this simple antibody test that people can take that can tell you whether or not someone has Alzheimer's, or at least increase the likelihood that someone has Alzheimer's. I should say that if you test positive, there is a much greater chance that you have Alzheimer's, is, I guess, the best way to describe it well. Anyways, abstract. After decades of Alzheimer's disease, or AD research, the development of a definitive diagnostic test for this disease has remained elusive. The discovery of blood-borne biomarkers yielding an accurate and relatively non-invasive non-invasive test has been a primary goal. So it's been a goal to have a simple test to give to people that can tell them whether or not they have the disease or not. Because if you imagine, the more complicated, the more difficult it is to do these, to diagnose, then you actually have to spend more money, more manpower, more energy in just trying to figure out what's wrong with people. But if you have a simple test that can do it very quickly, then you can get people treatment faster and cheaper, more affordably. Anyways, using human protein microarrays to characterize the differential expression of serum autoantibodies in Alzheimer's disease. So they used basically protein microarrays. So just I'll call it probably microchips. I didn't go too deep into the materials and methods, but there would be a way to buy products that have a laundry list of different proteins in the human body, and they can be organized in different trays. That way you can run experiments on a lot of different human proteins simultaneously, and you can do screening of a lot of proteins this way. So they used human protein microarrays to characterize the differential expression of serum autoantibodies. So they were looking for autoimmune attack on human proteins by taking their serum and running their serum. This might not be completely correct, but they were basically running the serum over these microarrays, and then the places where there were antibodies that stuck to those proteins, that would generate a signal that would tell the investigators, you have an antibody for this protein, and this protein is in your body, and it has this much signal. And so they can look at a screening of all of the human proteins, or a big number of the human proteins, and then they can say, well, this protein has a different signal than a healthy person. This protein has a different signal from a normal person. Oh, this one as well. And then you figure out which proteins, if there are autoantibodies for them, which proteins usually generate a signal, and then that can tell you that this set of autoantibodies are associated with one disease, and then perhaps you could have a different set of autoantibodies that are associated with a completely different disease. So, continuing, they were used to characterize the differential expression of serum autoantibodies in Alzheimer's disease and non-demented control groups, or NDCs. And then we identified potential diagnostic biomarkers for Alzheimer's disease. So the differential significance of each biomarker was evaluated, resulting in the selection of only 10 autoantibody biomarkers that can effectively differentiate Alzheimer's disease sera from non-demented control sera with a sensitivity of 96%. So if you are positive, it is 96% likely that it's going to positively identify you. And then it has a specificity of 92.5%. So if you do not have Alzheimer's disease, it will have a 92.5% chance that it will diagnose you as healthy or without Alzheimer's. So remember, nothing is perfect. And in fact, I should also mention that even the diagnosis done by a professional, even that isn't going to be 100% accurate. There's going to be some imperfect sensitivity and some imperfect specificity, even by a trained medical professional. So what might also be happening is we are making these tests using biomarkers to do a diagnosis, but perhaps the data that they were given might not have even been absolutely perfect as well. So we always need to consider everything is on a spectrum, and biology is a noisy field. So it is possible that that they got this data because the data that they were given might not have been perfect as well. Regardless, we should keep on going. Alzheimer's disease sera were also distinguishable from sera obtained from patients with Parkinson's disease and breast cancer with accuracies of 86% and 92% respectively. So that's pretty good. Uh, 9 out of 10 chance that it is a proper diagnosis, especially for two diseases that might have very similar clinical features. That would be phenomenal. Anyways, results demonstrate that and that's for the Parkinson's disease explicitly in comparison with Alzheimer's. Results demonstrate that serum autoantibodies can be used effectively as highly specific and accurate biomarkers to diagnose Alzheimer's disease through the course of the disease. So let's say, I don't want to go through all of this. So here would be an example of the identity and significance of 10 Alzheimer's disease versus non-demented control diagnostic biomarkers. And so here would be a list of different proteins. So here we have pentatricopeptide repeat-domain, or PTCD2. Here we have pro-opiomelanocortinin, which has a bunch of, what's it called? So it's POMC, and this has a bunch of other hormones that are located within it, such as adrenocorticotropin, or just corticotropin, or ACTH. There's alpha melanocyte stimulating hormone, and this is an important one, at least in the context of viruses, because viruses are actually able to create, or I should say, influence the production of autoantibodies against adrenocorticotropin. So if you have the flu, or if you have a type of coronavirus, you might actually be making autoantibodies against this. And if you could imagine, if you used this as your biomarker, someone with the flu might possibly test positive for Alzheimer's because they have more autoantibodies against adrenocorticotropin. That is just something to consider. So diseases can also influence your autoantibodies, is what I'm trying to get at. But we don't really need to go too in depth into it. Just know that they went into these and then they picked out the best ones. And in fact, are these the top 10 ones? Yeah. So these were the top. Oh, that's interesting. So let me 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Yeah. So these are the top 10 that they use. So fascinating. So the prevalence, yeah, let's go through this just so that you understand what's going on. So here, let's talk about the pro-opiomelanocortinin proprotein. And I'm going to use it because it is pertinent to today. Because today, we have a pandemic going around. And what is very likely going to happen is this particular virus is undergoing molecular mimicry of the adrenocorticotropin hormone. So ACTH, and I've done other videos on this as well, but basically what coronaviruses and some influenza strains can do, is they have figured out a way to cause, they do molecular mimicry of this particular hormone. And then when your body tries to fight off the virus, it sometimes will make antibodies for the mimic on the virus. And when an antibody is attacking the mimic on the virus, then it ends up attacking your own body's adrenocorticotropin. So what does this tell me? That if someone were to be infected with influenza and corticotropin, or influenza and or coronaviruses, which coronaviruses you do get infected with them, even the same species, multiple times throughout your lifetime, each time you get infected with one, it might end up being that it is increasing the amount of autoantibodies against adrenocorticotropin. If that makes sense. So with people with Alzheimer's, so here is the prevalence in Alzheimer's disease. 65.4% of people with Alzheimer's have detectable levels of antibodies against the pro-opiomelanocortin protein, of which adrenocorticotropin is one of them. So if you had a autoantibody against adrenocorticotropin, it would probably also attack the entire protein of the pro-opiomelanocortin molecule. But I believe this is usually cleaved immediately, and I don't think with their methodology they really could distinguish between antibodies between any of these peptides. Regardless, they have a antibody against at least one of the hormones that is within this POMC, or the pro-opiomelanocortin molecule. So people with Alzheimer's, 65.4% of them will have a detectable level of autoantibodies against one of these. And in healthy, non-demented controls, only 12% of them have detectable levels. So if you could, as you can see, there's a big difference. And here they have the p-value, which is the likelihood that this could arose from chance. And here it's very, very unlikely. So it's 1 × 10⁻⁵ of that happening. So that's 1 in, 10,000 chance that this is happening. So what they do with these diagnostic tools, though, is they don't just look at this one chemical, one autoantibody that's being produced. They look at 10 of them. And for each of them, there is a significant difference between them. Okay? And for each of those, if we, there might be some, what would the word be? Some correlation between them that isn't exactly perfect. But by combining the strengths of all of these, you end up with a diagnostic tool that has pretty effective specificity and pretty effective, specificity and, I hate sensitivity. You know, they try to do work with assonance to try to make everything sound nice, but it just makes things more confusing sometimes. So these were the 10 proteins that they looked at, or that they chose, because of how good of a difference there was in between the Alzheimer's group and the control group. So by doing that, they were able to create a diagnostic tool. So I am not going to go through the rest of the paper. I think the only thing that we should do is go over let's go over the conclusion. So, conclusion: the development of a reliable and accurate blood test for Alzheimer's disease will have profound clinical impact. The identification and use of a small panel of Alzheimer's disease autoantibody biomarkers shown here has a diagnostic sensitivity of 96.0 and specificity of 92.5 using available samples. And you know what? This is just the beginning. They probably have updated the antibody test now. They probably made it more accurate and more efficient by now, because this is already 10 years old. The relative non-invasiveness, low cost, and dynamism of protein microarrays make a diagnostic of this kind well suited for incorporation into routine healthcare. So this is a very standard procedure. So these protein microarrays, they are standardized, and a lot of health professionals across the world would probably be able to get these very easily, and probably, my presumption would be, it's probably relatively affordable because they might be mass produced. So take these protein microarrays, find a way to take a blood sample from someone to wash over them, and it can test for autoantibodies that way. And by testing for these autoantibodies, you can probably detect Alzheimer's disease, and probably many others as well. Now here, we hope that with a diagnostic such as this, accessible early screening methods can be established so that patients will be better positioned to avail themselves of effective therapies as they arise. So this is really important. Sometimes, well, these are progressive diseases. They're degenerative diseases. You degenerate over time with them. They progress, right? So one of the benefits of detecting these diseases earlier is that before you even show the worst clinical symptoms, if you can get a diagnostic tool, something in your blood is going to rat you out for whatever diseases you have. So if you can figure out how your body is speaking, then you can diagnose and you can start treating people earlier, and maybe you can prevent the disease from degenerating as quickly as it would have normally had it ran its course. Now, a few things that I want to think about that might be very important here is what's happening. Why are we generating autoantibodies in the first place with regards to Alzheimer's disease? So is it if you get Alzheimer's disease, your nerves are damaging, and through that inflammation or that destruction of cells, does that end up creating the autoimmunity? Or do we get the autoimmunity from somewhere else, and then that autoantibody ends up damaging the brain and causing Alzheimer's disease? And granted, for every one of these proteins, we could have that same chicken or the egg argument. Is it the or is it the lack of the firm domain containing 8 or FRMD8 autoantibodies? Are these autoantibodies actually protective that prevent Alzheimer's disease? Or is it if you have Alzheimer's disease, the level of these autoantibodies decrease significantly for some unknown reason? So same thing with the spleen tyrosine kinase. It appears as though healthy people have higher levels of that autoantibody than Alzheimer's disease people. Is it protective to have those autoantibodies, or is it after you get Alzheimer's disease, you lose the ability to produce them? Yeah, it's really hard to tell. And so for each of these, you could have that same argument. Is one causing the other? Is one causing the other? Or is it or is it just chance? But it's obviously not chance. So what I'm trying to get at is if there are diseases, oh my word, sorry, this is a completely different, this is the diagnostic test to determine between Alzheimer's disease and Parkinson's disease. So here are the 10 biomarkers that we were talking about earlier, and it looks like all of the biomarkers are higher if you have Alzheimer's disease versus when you are healthy and normal. So going back to that same argument, is it that people with Alzheimer's disease end up generating more of these autoantibodies as a result? Or is it if they have these autoantibodies in the first place, does it end up causing Alzheimer's disease, or at least making it worse? And could you say the same thing for every single one of these proteins? Well, for instance, this pro-opioid melanocortin, what would this tell me? It would tell me that it's possible that coronaviruses might actually be causing Alzheimer's. We already know that a lot of coronaviruses can cause significant, they can cause demyelinating events or autoantibodies against myelin, and they can cause nerve damage. So that might be one of the ways at which a coronavirus could cause Alzheimer's disease. So, and we do end up getting infected with coronaviruses over and over and over again throughout our life. And is it possible that coronaviruses are pushing us closer to Alzheimer's every time we get infected with them? Maybe only a subset of coronaviruses do that, or maybe a subset of flus do that. Who knows? But what I do know is getting these diseases is not good for your health in general. So it is possible that it might be better to just avoid getting them in the first place, because there might be unintended long-term consequences to it. So that is one of my fears right now, is knowing that there is a pandemic going on, and this pandemic might actually be playing a role in very long-term health consequences. That's something that I do not want to experiment with, especially for how deadly it seems to be in a subset of people. So if it's affecting some people very, very negatively, it's probably still going to affect you mildly and be doing something. You might have been better at preventing it from being as bad, but it's probably still doing something. So yeah, with all of that, I think we're good. So I want to keep today short. Well, let's do this one more thing, because this is my channel and I can do whatever I want with it. So let's go to Scholar and let's look up Alzheimer's, and let's do coronavirus. Let's see if there is a link anywhere. So vacuolating encephalitis in mice infected by human coronavirus OC43. Okay, we can just glance over that if we want. So here, let's look at what vacuolating encephalitis is. It could be something to do with the vacuoles of cells. I wish. What is the formation of vacuoles or vacuole-like structures within or adjacent to cells. So the formation of these vacuoles is causing encephalitis, or it's causing inflammation of the brain in mice who are infected by human coronavirus OC43. So this is one of the normal human coronavirus, one of the 4 that circulate around year after year. I shouldn't say year after year, but faster than a year after year. It's always happening. It's endemic to the human race. Oh, really? So human respiratory coronaviruses can infect neural cells and persist in the human brain and activate myelin-reactive T cells. Well, yeah, that's what I was talking about earlier, was some coronaviruses can cause autoimmunity against myelin or against the nerves. So that's interesting. And this, as a means of understanding the human infection, we characterized in vivo the neurotropic and neuroinvasive properties of HCOV-OC43 through the development of an experimental animal model. So virus inoculation of 21-day postnatal mice led to a generalized infection of the whole central nervous system, demonstrating HCOV-OC43 neuroinvasiveness and neurovirulence. So let's look up HCOV-OC43, what receptor it uses. I think, is this the one that was associated with Kawasaki's disease? I love Wikipedia. It's great. It infects humans and cattle. Oh, it binds to the N-acetyl-O-acetylneurominic acid receptor. So this is one of the receptors that's probably very expressed on the neurons, but it's probably, I think it's expressed everywhere, but you know, it's hard to remember because there's a lot in biology to remember. So This residue is negatively charged at physiological pHs and is found in complex glycans on mucins and glycoproteins found at the cellular membrane. New 5-ACK residues are found in glycolipids known as gangliosides and are a crucial component of neuronal membranes found in the brain. So it seems as though it is predominantly within the brain that you find these, which makes sense that HCUV OC43 is able to infect neurons. So, and then persist in there. So remember, your nervous system is possibly an what would the word be? Oh shoot, I forgot the word. I did a whole video on it. Immunoprivileged. So it might be an immunoprivileged area, which means that the immune system does not function as effectively or as efficiently in those areas. Probably to try to prevent things like this myelin reactive T cells from destroying your brain. So having your immune system suppressed in the brain is an important thing for health. Anyways, this acute infection targeted neurons which underwent vacuolation, or the creation of vacuoles, and degeneration. So these vacuoles, I assume, just kept on growing and the cell just died. While infected regions presented strong microglial reactivity and inflammatory reactions. I believe the microglia are immune cells within the brain, but yeah, so it is one of the few immune cells that are within the brain. So damage to the central nervous system was not immunologically mediated, and microglial reactivity was instead a consequence of direct virus-mediated neuronal injury. So here, okay. So here they're saying this is not an autoimmune disease. They're saying this is directly from the damage that is done by the virus. So it's not, the damage isn't being done by the immune system, the damage is being done by the virus. So that's not exactly what I want to look at. Neuroprotective effects of anti-lipoprotein D. Let's look at the since the year. Yeah, it looks like now everything is about COVID-19 if you haven't updated enough. So I think I will leave with this then, because I don't know if I'll be able to just stumble upon a good paper within the next few minutes. So thanks for watching. Hopefully I will have another video up by tomorrow. And if you have any questions, leave them down in the comments, or any corrections, you can leave those down in the comments below with all the bots. So anyways, y'all have a good one, and I will see you tomorrow.